Today, I'm going to briefly go over what forensic toxicology is because that's the space within which I do research. I'm going to cover the state of practice in the forensic tox world in Wisconsin, including the workload at the Wisconsin State Laboratory of Hygiene and some data on trends in drug detection. Then I'll show you results from a roadside drug detection project we carried out, and I'll finish by going over cannabis, highlighting the biggest knowledge gaps and most pressing challenges in the cannabis consumption space from my perspective. So I like to put acknowledgements at the front because they're really, the people are what helped me get all of this together. It's not a one-man band for sure. So I've mentored 4 undergraduate pharmacology and toxicology students. 3 have recently graduated, and I have 1 chemistry PhD student, Maya, who's in the picture in the teal dress. And without them, none of this would be possible. So I like to acknowledge their contributions. And also I work alongside the Wisconsin State Laboratory of Hygiene's forensic toxicology section staff, who are instrumental in gathering and sharing their data with me. Also, as practitioners, these forensic tox staff are great resources for identifying current pain points and knowledge gaps in the forensic tox space, which helps guide my research proposals and ideas. And then second only to the people is the financial support required to make this work. So the roadside impairment detection study I will talk about today was funded in part by Wisconsin's Bureau of Transportation Safety and Technical Services. The cannabis impairment pilot project that I'll discuss at the end of the presentation was funded by UW's Institute for Clinical and Translational Research, or ICTR. And finally, we have funding from the National Institutes of Justice for another project that I won't be discussing, but the funds are the graduate student Maya Bates, who's contributed to some of the data presented today. These are the learning objectives for the presentation, and I'll use this to orient us and myself to where we are in the presentation as we go. So we're going to start by just defining forensic tox and creating a general awareness of what Wisconsin's impaired driving laws are in the state of practice, just so we're all on the same page. And I also like to put in a plug for the Wisconsin State Laboratory of Hygiene. As I've joined the UW and interacted with people, a lot of them ask what exactly the state lab does. So I want to just explain that now. The Wisconsin State Laboratory of Hygiene is Wisconsin's public environmental and occupational health laboratory, and I hold a joint appointment with it, and it's where my lab space physically is. It's part of UW-Madison. It's an auxiliary unit reporting to SMPH, so we're kind of colleagues. And it utilizes analytical services, research and development, public health programs, laboratory proficiency testing, and training and education of students and professionals to carry out the labs and the university's mission. And then this is just a recent organizational chart to give you an idea of the scope and breadth of services offered by the Wisconsin State Laboratory of Hygiene. And you'll notice that there's a communicable disease division, disease prevention, and then I reside within environmental health in the forensic tox section. So this is a definition of forensic toxicology, which is really any application of science and study of poisons to things that relate to judicial proceedings. And basically, I think of it as a mashup between analytical chemistry, pharmacology, and the law. And then this is a very simple representation of the tasks performed in forensic tox, but I really like how clearly it boils down what we do. So when submitting, the submitting agency is law enforcement, we do the same types of tests, but the end product is often testimony at trial. And when coroners or medical examiners submit samples to us, they're interested in the relevance of any detected compounds to the cause or manner of death. And then forensic toxicology in Wisconsin is carried out by 3 distinct agencies, which further complicates the picture. The Wisconsin State Laboratory of Hygiene, which is where I work currently, The Wisconsin State Patrol has a breath alcohol toxicology program, which I managed for several years prior to joining the hygiene lab. And then the Wisconsin State Crime Lab, which covers a wide variety of different cases. And going into where specimens go, the hygiene lab will take all impaired driving offenses and does, quite frankly, most of the impaired driving casework for the state of Wisconsin. We will also do death investigations, whereas at the crime lab, they will only accept samples if they're associated with a felony charge. And then the chemical testing section, just due to the limited nature of breath alcohol testing, they can only do breath alcohol investigations. This is from a community maps of crashes that have occurred in Wisconsin where a driver was suspected of or cited for impaired driving, and each dot on this map is a unique crash, and the color of the dot indicates the severity of the crash. So an injury that is incapacitating is where that person is not able to walk, drive, or perform other activities, like a broken leg or unconsciousness. Non-incapacitating is where they've received perhaps a laceration or some contusions that are visible, but they can still move around. And then a possible injury is something that's not immediately evident to an observer or first responder, but that patient or victim claims they have, like maybe whiplash or some other soreness. So that gives you an idea of just the type of severities, and then just the profound number. So I queried this on about a week ago, and there were already over 2,500 crashes, 63 fatalities, and 1,200 injuries associated with impaired driving. And then these are the Wisconsin's impaired driving statutes. I only bring this up because they're kind of complicated to navigate, and if someone is cited for impaired driving, they'll often get cited for several things that mean the same thing or very similar things. So when it goes to trial, it's kind of a prosecution strategy. But impaired includes, so the first bullet point, any intoxicant, controlled substance analog, or other drug where they may be unable to safely drive. So this also includes any detectable amount of a restricted controlled substance in their blood or a prohibited alcohol content. So the first bullet here, illegal to drive while impaired, is the most encompassing, and then there are several subcategories that are more specific. And then also just to cover our statutory alcohol limits, because I'm always surprised. I work in this space, so I take for granted that these are hard facts that everyone knows. But I guess if you don't work in the space, you wouldn't necessarily know. These are, so 0.08 is the hard and fast per se limit for all states except for Utah, who has 0.05. And then depending on if you have a safety sensitive position or if you're not supposed to be consuming alcohol, like you're under 21 years of age, there are different rules that apply. And also of note, if you're pulled over and you have an alcohol concentration of a 0.15 or higher, so almost double the legal limit, you can receive an enhanced penalty even on your first impaired driving offense, which is the utilization of an ignition interlock device to resume driving. These are the Wisconsin's restricted controlled substances, which is anything that's Schedule 1 under the Uniform Controlled Substances Act, or an analog of those substances, or cocaine and its metabolites, methamphetamine, and then delta-9 THC is also specifically called out at concentrations of 1 or more nanograms per milliliter of a person's blood. We do what's called implied consent testing, which states that anyone who operates a motor vehicle on Wisconsin's roads is deemed to have given consent to a search of their blood, breath, or urine for the presence of drugs and alcohol. So that's the law within which a lot of our blood specimens are collected. For our impaired drivers, we do only blood analysis. We don't consider urine or other oral fluids for the generation of evidence for use in testimony or trials. Drug testing must be requested by the submitting agency. So if you just send us a sample, we're not necessarily going to run it through all of the workflows. And then we have a cancellation policy where if we observe a blood alcohol concentration of 0.10 or greater, with a few exceptions, we will cancel testing, which if you recall from a couple of slides ago, there's enhanced penalty for a 0.15 blood alcohol concentration. So that seems a little counterintuitive that, we have these laws in place for enhanced penalties if you're, there's a perception that you're very impaired or you have a very high BAC, but then due to resource limitations, we have this cancellation policy. So it's assumed that we're missing a lot of substances in these higher BAC drivers. And then also, if a restricted controlled substance drug is detected, further drug testing is canceled, and the thought process is it's a restricted controlled substance by law in Wisconsin. So if it's on board, we don't need to waste resources combing this sample for all other substances. But then this also creates a gap in their knowledge of the prevalence of polypharmacy or polysubstance use in impaired drivers. This is our workload graph going from 2014 through 2021. And what I like to draw the attention to is the increase in the relative amount of drug tests as it relates to total samples, which is what the line graph is showing. So there's been a substantial increase over these past few years in the amount of drugs detected in our overall casework. It used to be a lot of alcohol and then drugs were fairly rare, and now it's seeming to be more of a 50-50 split. And even more so, considering this is including our drug cancellation policy of the higher BAC, so there's likely an even higher amount of drug prevalent that we just don't have the resources to fully quantify and find. And then I always like to show this slide. Second only to alcohol is THC as our most prevalent drug in Wisconsin. And following that is methamphetamine and amphetamine, benzoylecgonine, which is a metabolite of cocaine. And then coming in fourth is fentanyl. So that's just how prevalent fentanyl is in our impaired drivers in Wisconsin, which always really, it blows my mind because it's just so potent, but it's seemingly to be, omnipresent. And then, like I alluded to before, a thing that we're seeing a lot more often in our cases is polysubstance use. And this isn't necessarily that the person who's using the substance is seeking out multiple substances, but there's also reports of a lot of polysubstance use from contaminated or tainted or cut substances they're buying on the street. This comes from a publication from last year out of the lab, but what it's showing is that even at higher blood alcohol concentrations, the amount of drugs on board, 1 or 2 or more, is still a significant percentage of those cases. So again, going back to, what is the loss of data from our cancellation policy, but also, how do we identify impairment when it's not a clear-cut one substance? The people are under the influence of several competing substances at the same time. So moving on, now I'm going to talk about the strengths and limitations of existing driving impairment detection strategies. The first and most common is standardized field sobriety tests. These are very effective in detecting impairment, but additional observational tests might be needed. These were designed to detect alcohol impairment, so they really, quite frankly, fall short when people are under the influence of several compounds or drugs and not alcohol. And then the officers need to be prepared to present this observational evidence in court, which is pretty tricky because, my observations versus yours may be somewhat different. And while they're very trained in as much standardization as possible, these are being conducted at the roadside, not in a laboratory setting. So there's a lot of competing interests for that officer's attention in a variety of different situations where the tests are conducted. Then there's special training that's offered to officers once they've shown mastery of their standardized field sobriety tests. So these are additional psychomotor and cognitive tests that are also conducted at the roadside. And this sort of extra training is, we'll talk about a little bit more later, but they're called drug recognition experts. This can be very effective in characterizing impairment, particularly from multiple different classes of compounds. But many jurisdictions have too few drug recognition experts, and the drug recognition test takes about an hour to complete because there's so many different tests that need to be done. And then they are expensive because there's a lot of training that goes in, and these officers are required to do recertifications and prove that they're maintaining proficiency in the space. So it's a long and ongoing maintenance of their credentialing to be a DRE. Then there are roadside chemical tests. The most prevalent is the preliminary breath test, which you may have seen in movies where people are asked to blow at the roadside to determine just approximately what their blood alcohol concentration may be. And there are emerging technologies for doing that same thing using oral fluids, which I'll talk about, but this can be helpful in informing the decision of impairment, but no test will cover all drugs, and a negative roadside test does not necessarily rule out impairment. So this is more a tool in the kit that's kind of supplemental to the prior two tools. And then laboratory-based tox testing can provide powerful evidence, but there's a lot of issues with delays, backlogs, insufficient training to interpret data, insufficient scientific evidence to interpret data, which I'll talk about in the cannabis space. And then this information can't be used to establish impairment. It's just kind of, again, supportive information, but that officers' observations of the impaired driving behavior is really critical for this all to go. Standardized field sobriety tests were developed in the 1970s. And again, like I said, it was focused on alcohol impairment. It includes a one-leg stand, which it is a divided attention task. They have to balance, count, and follow instructions. A walk and turn, where again, they're walking and following instructions. And then horizontal gaze nystagmus, or HGN, which is an involuntary jerking of the eyes. Drug recognition experts carry out a drug evaluation and classification, which includes several more tests. In addition to HGN, they look at vertical nystagmus and lack of convergence, pupil size, a modified Romberg balance test where they're looking at that person's internal clock and ability to keep their body oriented in space, finger to nose, and vital signs, which is why this takes a lot longer to complete at the roadside. This is a video, and I'll show you, but it's just showing what horizontal gaze nystagmus looks like. There's no audio for it, but it's just helpful to see that eye twitching when I'm talking about all this, so we know what I'm talking about. So you'll see the eye kind of bounce a little bit as it gets farthest off to the side as they're following the officer's stimulants right there. So that's what the officer is looking for. And then moving on, this is an example of lack of convergence, which is where the eyes don't move together. So you'll see here, one eye kind of floats off to the side where the other one stays in the middle. And that subject thinks that they're observing that their eyes are in the same place. So those are just helpful images of what they're looking for. And then moving more into the biological specimen space, which is where I do a lot of research. There are several specimens that you can collect to use as evidence of impaired driving or to look for clues of substance consumption. Blood is the most common, but oral fluid is increasing in its popularity. And then I like this figure because it demonstrates just kind of the time frame of by the biological specimen what sort of recency you're getting from that, which is, and then it becomes evident why blood and oral fluid are so popular and urine is less popular in this space. And this is just the publication that I'm going to talk about here, but we wanted to explore alternative matrices, specifically looking at the utility of surveying oral fluid versus blood. So once there was a suspected impaired driver and they agreed to, participate in this study, the whole blood specimen was collected routinely, but then 2 additional specimens of oral fluid were captured. One went into a lateral flow immunoassay device, which was done at the roadside and gives immediate drug class feedback. And then we also captured an oral fluid specimen for laboratory analysis, which we thought we could more directly compare to the blood sample because we ran them on the same instrument using the same assay. And then we compare the results of these 3 approaches to the drug recognition expert evaluation when they were available. This study included 106 participants. They were primarily male, a wide age range, although the median was 32. Most of the times of arrest occurred in your stereotypical impaired driver times of 8 p.m. to 4 a.m. And then almost 80% of the blood specimens contained alcohol, whereas just under 70% had greater than a 0.1 gram per 100 milliliter concentration of alcohol. And then this bar graph shows the distribution of blood alcohol concentrations observed in this study. And then this is each bar here, each color, the red is the roadside oral fluid, orange is blood, and then the yellow is the oral fluid sample run in our laboratory assay. And this shows that no test and matrix is perfect because there are clear limitations, particularly for THC, where THC, being a highly lipophilic compound, doesn't partition into the oral fluid very well. So we didn't observe a ton of oral fluid in the oral fluid tests that we did in the lab, whereas it was observed on the oral fluid collected at the roadside, and that may also be because our target compound was different. At the roadside, it was looking for the parent delta-9 THC, whereas in our laboratory assay, we look for the carboxy metabolite. So it's just a complicated picture, and nothing is going to be a one-size-fits-all approach for roadside detection. When we compared the roadside device to blood, this is kind of a, not super helpful table for you all, but the takeaways are that for amphetamine and methamphetamine, there were several positives via the roadside device that did not have a matching blood positive. And the detection of benzodiazepine drugs in oral fluid is challenging because it's a diverse class of compounds of variable potencies and low concentrations in the oral fluid because these compounds tend to be protein bound and lipophilic. And then we also observed some cross-reactivity with the assay in the roadside test. The Sotaxa roadside test cutoff concentration for cocaine was lower than our test in the lab, so that created some discrepancies. And then we found most concerningly is that the Sotaxa was not sensitive to fentanyl because at the time of its deployment, fentanyl wasn't a big thing in the recreational use space. But now, as you saw from my top 10 list, it's a predominant opioid. So over time, these roadside screening tools may lose utility as the drug use landscape shifts. And then for THC, the Sotaxa detected delta-9, whereas blood, we looked for the carboxy metabolite. So again, that creates some discrepancies because you can have delta-9 THC contributed to your mouth oral fluid mucosa from just being nearby other people that are consuming cannabis, particularly if it's vaporized or combusted. So if you're in a car and there's, secondhand cannabis smoke, whereas the carboxy metabolite is only going to be present if you've actually consumed and metabolized a significant amount of the parent compound. So that's another limitation with the oral fluid, is that there can be contributions from orally delivered via either inhalation or just, chewing and consuming or swallowing to that space potentially without actually getting the impairing effects of those compounds. And then another goal of our study was to assess how drug use trends in Wisconsin changed over time and how that impacted our detection. And there was a previous study done by the hygiene lab in 2016 in Dane County. And the demographics of Dane County I like to include because they're so different than Manitowoc. So there's over 500,000 people. The inhabitants per square mile is a little bit more compact, and it's a bit more diverse than Manitowoc County, which is where the study that I just referenced was conducted, where it's only, under 90,000 people, far more rural, more spread out, and predominantly white population. So the drug trends, comparing the drugs observed in Dane County in 2016 to the drugs observed in Manitowoc County in 2019, there was an increase in opioids, and that may be due to the time. And there was a decrease in benzodiazepines, but an increase in methamphetamine. So, there's too many variables at play here to really discern if this is a geographical or a time change in these trends, but it really exemplifies how these things shift over time. Then we looked at the drug recognition expert evaluations, and I'm not going to go through them in detail, but the take home is that most matched all toxicology reports, and then several matched one, but not all of the toxicology reports, and only 3 of the 21, so 15% did not match. And in those 3, depressants were called, but no drugs were detected. Another one was that the DRE suspected depressants, but the person had SSRIs in their system. And then, there may be that if they were undertreated for depression, or that they were, still feeling the effects of depression, that there may have been some impairment in their driving. I know if I drive and I'm in a bad headspace, I can be distracted. And then another case, the DRE thought it was depressants, narcotic analgesics, and stimulants, but then only stimulants were detected. So this is, identifying, like I talked about, the strengths and weaknesses of these different approaches and how you kind of need a holistic approach with multiple different data elements to make a clear, case about what that person, how they were acting observationally versus what was in their biological specimen, and then knowing the limitations of the different tests utilized to do those analytical assays. Because just because we did our assay and didn't detect anything doesn't mean that there wasn't something there that just isn't in our scope of testing. That's also very possible. Then moving on, I'm going to start talking a little bit more about cannabis impairment. I like to start off by just laying the foundation of what a cannabinoid is and why do we care. So there's over 100 botanically derived phytocannabinoids, and many directly impact our endocannabinoid receptor system, but it has a diverse pharmacology, and on top of that there are hundreds of synthetic cannabinoids. Our endogenous cannabinoid system is essential for critical survival functions, including hunger, regulation, body temperature, memory, fear, and reproduction. So therefore, it's tightly controlled and rapidly changing, and we have endogenous and cannabinoids or endocannabinoids shown below, and as these bind to our receptors, they can trigger second messenger signaling pathways in response to external and internal stimuli, and the endogenous cannabinoid system attempts to maintain homeostasis of our bodily functions, including neuroadaptations to the brain, which is the problematic point of this if you're consuming cannabinoids while pregnant or breastfeeding or still developing as a youth. And then this is a nice figure I like to show because it kind of displays where all of the CB1 and CB2 receptors are in our body, and then explains why we see a diverse impairment profile from cannabis and cannabinoids because they are so prevalent in the brain, different parts of the brain. There are physiological, cognitive, and motor effects. A lot of these are things that the drug recognition expert examination is trying to quantify, but it also impacts your ability to do safety-sensitive jobs or drive a car. So it affects your executive function, your attention, concentration, memory, decision-making, etc. And then also motor effects with planning and implementation, reaction time, tracking, stuff like that. And then as society moves towards decriminalization and legalization of cannabis and cannabinoids, either for medical or recreational use, there's a wide variety of different forms of cannabis. So I like to show this slide because oftentimes these words and descriptions are moving into the scientific literature. So I often have to Google, what does a shatter mean? Because it's not something that I was taught when I was in college. So maybe it wasn't really available when I was in college, but it's the project we're working on that I'll get to in a few slides. We're using a distillate of delta-9 THC because we're hoping to try to identify the impairment profile without the potential effects of the other cannabinoids or compounds present in the cannabis plant material. This is the chemical structure of delta-9 THC, which is the primary psychoactive ingredient of cannabis. And once you consume it and metabolize it, your body converts it to the hydroxy form, the 11-hydroxy form, which is also psychoactive. And then it goes again to the carboxy form, and this is when it's no longer psychoactive, and this or its glucuronide conjugate is what's excreted in the urine. And oral inhalation versus oral ingestion causes a vastly different pharmacology, and that's due to this first pass metabolism. If you inhale your cannabis or THC, you will have pharmacodynamic effects within minutes, a peak of after 15 to 30 minutes, and it will resolve after a few hours. Whereas if you have an edible, for example, you're not going to begin to feel the effects of that for 30 to 90 minutes because your body has to convert that delta-9 THC to the 11-hydroxy THC before it circulates in your blood. And then you're going to have a peak maximum after a few hours and resolve up to 12 hours later. So you can see these are very different time courses for the same compound, or the same compound that you're ingesting at least. Also, the bioavailability fluctuates. So how much of a consumed THC product actually makes it into the bloodstream? That's not an easy, there's no easy answer to that seemingly straightforward question. So in this experiment, there were 6 test subjects that smoked the exact same amount of delta-9 THC. And then you can see that there's one test subject that had almost 3 times greater peak than another. So for all test subjects, the peak occurred at about the same time, but the relative amount of THC in their system varied wildly, which is important to know because a lot of jurisdictions that have chosen to decriminalize or legalize cannabis consumption for adults in their jurisdiction have also put out per se limits on the amount of THC you can have in your body when you drive, just like alcohol. But the compounds of alcohol versus THC are so vastly different that that's not based in a scientific principle. There's no data supporting a per se limit for this exact slide. This graph is why any per se limit you see is just kind of nonsensical, because people can consume the same amount and have wildly different levels. So then the next natural question is, what's influencing this? And the two main factors, at least as we understand it today, is the method of administration, so inhalation, and that includes your inhalation pattern. So people that consume cannabis regularly will become more efficient at inhalation administration. And then whether they are an infrequent or regular user will also impact that, due to things like their puff duration, if they hold their breath, etc. So regular users will have 50 to 70% higher bioavailability than an acute user. And then, like I just mentioned, higher THC levels is higher person, and that is false. Then the THC impairment versus biological concentration profile. So in blue is the subjective high, and then purple is the performance quantification, and then red is the THC concentration. So you can see that there's this peak and then rapid fall off as the body's metabolizing THC, but you're having this prolonged subjective and quantifiable performance decrements in these individuals without the corresponding high THC biological concentration, which is very problematic in my forensic tox space. And again, due to the how lipophilic THC is, it has different concentrations in various tissues. So in the fat, it bioaccumulates over time, and therefore you can detect it there for a long period of time, but that's not a great specimen for determining if someone's an impaired driver. Yeah, so this is just all kind of culminating in that it's nearly impossible to identify a minimum concentration of delta-9 THC that is consistently associated with impairment. And everyone's looking for this one magical number. And unfortunately, I think it's just not, it doesn't exist. So occasional users may be very impaired at a very low concentration, whereas chronic users may have over 1 nanogram per mil, which is our cutoff in our lab, for weeks, even once they've stopped smoking, because of bioaccumulation effects. So it's a very complicated landscape. Cannabis impaired driving has a few hallmarks. If you've watched any just TV shows or movies, it's often, you're stopped at a red light, but it's a stop sign. So that's sort of like impaired judgment, impaired ability to pay attention to your surroundings. Oftentimes these individuals are going too slow and experiencing lane deviation and inability to maintain following distance. When we do impaired driving tests, we look for the standard deviation of lateral position, which is a measure of weaving of the vehicle within the lane. And I'm not going to go over these in detail, but the most reliable indicators of impairment from the drug recognition expert perspective is kind of a culmination of nuanced observations, which makes interpretation very challenging. Moving on to more challenges in the cannabis research space is THC isomers. So I did a cursory review of our data, and about 5% of the samples tested at the lab in the last few years that contain delta-9 THC also contain delta-8 THC, which is an emerging isomer. And the chromatographic separation of these isomers in our assay is very different, because I'll show you in the next slide. The difference in the chemical structures is the shifting of a double bond. So it doesn't give us a lot to work with in terms of how to pull these compounds apart. Also, strain names as advertised are effectively meaningless. So cannabis sativa or indica, they're all hybrids. So those are all just kind of marketing ploys from my perspective. When doing research, we need a cannabinoid inventory to full mass balance to have any impact on interpreting the effects of different cannabinoids and phytochemicals in that compound. And then we also need to be wary of operationalized terms like isolate, full spectrum, et cetera, and just being very careful in identifying what that means. And then there's a large gap between the science and retail availability or medicinal use claims, and this gap, although already wide, seems to be widening. And then the representation in clinical trials is also a challenge, meaning demographically representative of people who use cannabis. Speaking of THC isomers, after the Farm Bill was put into place, people all jumped on board and started mass producing a ton of CBD. And then the price of CBD fell out because everybody was producing it. So the market was flooded and people needed to figure out what to do to recoup costs. So with a very simple chemical conversion, you can convert CBD into delta-8 THC as the primary product, but also delta-9 and delta-10. And if you pay attention to where this double bond is moving, that's the only change in these chemical structures. So you can see why they're so challenging to differentiate. And this is data pulled from the National Survey of Drug Use and Health, showing the shifting trends in cannabis use. And I like to start with the good news, which is in the 12 to 17-year-olds, is there's an overall downward trend. But then, you know, there's an increase in particularly the older Americans in their utilization of cannabis. And this is past month, so it's an indicator of recency of use, with particularly the 65 plus increased over 2,000% from 2005 to 2021. So that's a big step up. And then there's also a gap in clinical trial participant demographics. So this is the result of a literature review compared to that same National Survey on Drug Use and Health data. And you can see the discrepancy where clinical trial participants are overwhelmingly white. And also there were issues with the age. They're often 30-something white men in these clinical trials, whereas the people who use cannabis are an increasingly diverse cohort of people. And that gap can be very problematic for translatability of any results from the clinical trials. And finally, I'm going to briefly go over the regulatory approvals process required to initiate a human subjects project involving controlled administration of a drug of abuse, particularly a Schedule I. This is on our pilot project, which we've called High Confusion. We aim to evaluate the feasibility and acceptability of administering delta-9 and delta-8 isolates using standardized dosing units and quantifying the pharmacokinetics, and then also evaluating the driving impairment compared to a placebo. This is a double-blind, placebo-controlled crossover study design, and since it's a pilot, our N is 5. Again, these are the chemical structures of delta-8 and delta-9, and our study, each study drug administration session includes a lot of different data collection, which I guess is why it's great to do the feasibilities. We can see if we can pull this off. But we're going to collect a ton of biological specimens, including blood, oral fluid, dried blood spots, vitals. We're going to have a collaboration with the mechanical engineering faculty and do a driving simulator study using their full-scale driving simulator, which is a picture of it is here. We're doing some cognitive tests that are computer-based. We're inviting a drug recognition expert to come in and run them through a full eval just to see exactly how all of these different measures of impairment actually work. Navigating the regulatory approvals process is challenging, particularly as this is my first clinical trial, so I jumped right off the deep end into cannabis administration. So far, it's just been a ton of paperwork. We're nearing the end of our regulatory approval process, but to get a little bit more into the weeds of what exactly goes into this, because when I started looking in the space, I always wondered, you know, why don't we do more research on humans that would really clarify things quite nicely? Well, it's not so straightforward. So the first step is you have to get the FDA to allow you to do the work. You have to have your full protocol developed. and you have to work with a vendor to get the chemistry and manufacturing controls information for your active pharmaceutical ingredient. That's what's required for your investigational new drug application. Once they've green lighted things, then you have to go through your IRB. So if there are any amendments that your IRB wants, you have to loop back with the FDA. Once the IRB has approved your protocol, then if you're doing this work in Wisconsin, you have to go through Wisconsin's special use authorization for utilization of a controlled substance, which is like a mini DEA. And then you have to go to the actual full DEA to get approval. And that's all before your trial can actually enter active status and you can start recruiting and enrolling participants. So like I mentioned, if any step along the way, any of your regulatory approval bodies need any adjustments to the protocol, you're going to have to go back to the beginning and submit amendments sequentially to all of the oversight bodies to get approvals to go forward. And then once you've gotten the green light from all of those agencies, then you get to actually do the work of enrolling your participants and seeing how the pilot study goes. So that's where we're at. We're waiting on DEA approval and final FDA clearance once the DEA gives us our license.